All right, in this video, we're going to focus on now kind of the details of DNA replication. So the title of this section says, the parental strand is used as a template for the formation of a complementary daughter strand. I'm a big fan of highlighting things. So if I highlight what we are identifying as the parental strand, we mean that these two original pieces that we've separated. And then if I use yellow to represent a daughter strand, we're talking about the new strands that we are forming. Okay. So it says down below that DNA polymerase is going to add nucleotides to each growing strand. So remember that here's your primer, right? We had to place that RNA primer. This was the 3' end of that nucleotide. So new nucleotides have been brought in by DNA polymerase. We didn't show it in here because we didn't want to clutter up the picture too much. But the newly formed daughter strand is complementary to the parent. So if the parent has a base of A, then the daughter strand would have T. If this is a C, then this is a G, so on and so forth. Okay. DNA polymerase continues toward the 5' end of the parent, right? So we're talking about 5' end of the parent. So if I labeled this 5', sorry, maybe I'll switch to black so you can see it a little bit better. Okay, that's the parent. Notice that this is still 3' on the daughter strand, and that's what allows more nucleotides to be added, elongating in this direction. Now, when we look up at this side, we start to see something that could potentially be a problem. If this is the 5, right, then here's the 3 on the daughter strand. And then if this is 3, this is 5 on the parent. Okay, so replication is happening in this direction. Okay, so we start to see that it's happening in 2 directions. Let's look at this a little bit more. So this is the same thing that we were looking at just a second ago. Remember that helicase is breaking these bonds and opening up the DNA in this direction. So that means that as new nucleotides are being added, this daughter strand being formed could continue on following right behind helicase without any interruption. Okay. On your notes, it says one daughter strand will be replicated continuously behind helicase. So this is known as the leading strand. However, on this side, we placed a primer. We added new nucleotides to that 3' end, so replication happens in this direction. But what about all this space? Well, we have to place another primer. And then we add nucleotides to fill in all the way over here, right? But remember, as helicase continues, then you're going to open this up even more. So then I have a gap. So because DNA polymerase can only attach nucleotides to a 3' end, this is called the lagging strand. And it's formed in small fragments known as Okazaki fragments. So what has to happen is, as helicase continues to unzip the DNA, RNA polymerase or primase has to keep placing primers so that that side, the lagging strand, can continue to be formed. So when we look down here at this, these red pieces are those primers, the RNA primer. Okay. The blue would be newly formed, right, DNA. So it continues on, but eventually you'd run into the next primer. So if I go back up to this picture, see if I continue this on, if this is building, 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 eventually it runs into this primer that's already there. That piece is already there. So each of these little pieces are known as Okazaki fragments. So what has to happen is, it says the RNA primers are later replaced by DNA nucleotides by yet a different DNA polymerase. So DNA polymerase 3 is the one that's adding the new nucleotides, elongating the DNA. DNA polymerase 1 is actually going to take out the primer and then put in DNA nucleotides, right? And then thirdly, yet another enzyme, DNA ligase, is going to seal this gap in between by creating that phosphodiester bond. Remember, that's that strong covalent bond that we use in the sugar phosphate backbone. So as a result, each new molecule of DNA, so the molecule is the double helix, consists of one original parental strand and then a newly synthesized daughter strand, because DNA is double stranded. When we did our science practice, we identified that Meselson and Stahl figured out that this is known as semi-conservative replication, okay? That your daughter strand would contain, so these are the daughter strands, would contain one old and one new. If we thought about this like what we did in our notes, the parental strand we had as blue, and then we drew new DNA in as red. So it would be like tracing over this with that blue and red. Sorry for the purple. Didn't mean to confuse you there. Okay, so that's semi-conservative replication. Both of these, this is a molecule of DNA, this is a molecule of DNA. We're saying that these are the newly formed pieces of DNA. The parental would have been like all blue. All right, almost done with this part of the video. So a few clarifications. There are many replication origins within one chromosome. To specify here, we're talking about eukaryotic DNA. Okay. So like we said, there's the bubble, there's the bubble, there's the bubble, and they each have their own fork. Okay, so they can replicate and then they run into each other and then the whole thing gets replicated. So that way it happens super duper fast. Okay. This is a highly simplified representation of replication. There are some steps and some enzymes that we don't need to worry about, and that's totally fine. There are still a lot of questions and things that aren't clear exactly about how it works. We do know that there are some enzymes that proofread our DNA for errors. There have been, up to 130 of these kinds of enzymes identified in humans. So there's lots of things that we're looking for or checking for within ourselves. So this is just a specific kind of a did you know side fact that there's a type of nuclease enzyme that's involved in nucleotide excision repair. So this helps repair DNA that's been damaged due to UV radiation. When you look at this, when the sun shines, sometimes it causes these thymines to bond together and kink this DNA like this. So it's kind of messing up those hydrogen bonds in between. So in this particular disorder, xeroderma pigmentosum, they lack this repair enzyme. As a result, what do you think these people would be more prone to? Probably skin cancer. All right, I'm going to stop the video here. There is one more video looking at telomeres, and then we're almost done.